Well, let's kick off the workshop with a classic disclaimer. So today I'm here. My flight and my hotel fares are paid by Kaijin, and I also was paid early this year to the ESCO meeting. However, I was not paid enough, like my boss, who will be here in a couple minutes, Dr. Andrew K. Godwin. He's the director of our clinical molecular lab. And also, surprisingly, our pathology laboratory director, Ada Persons, she is here too. So I can guarantee you this is going to be our scientific discussion. And today, the topic is implementation of next-generation sequencing for molecular profiling in a CLIA CAP certified and accredited laboratory. So first, I want to share you with one example why I think it's awesome to have NGS testing developed in-house. So most of us know that on August 1st, 2017, there's a new drug called enasidenib. It's approved for refractory acute myeloid leukemia and for relapsed AML as well with an IDH2 mutation. This drug is so awesome because it has no cytotoxicity and it has real tumor lysis syndrome. What's more is it's a daily oral drug, which is much better than other chemotherapeutic compounds. And this drug does have a little bit side effects. However, it's either reversible or it's treatable. And on August 2nd, we were able to search our internal database, and we were identified 24 patients who have IDH2 mutations. So on the second day after the FDA approved the drug, we were able to give this list to our physicians, and then they can take it from there. The physicians are really impressed by what we have and what we can provide in such a timely manner. If you think if you're only doing the regular reflex testings for AML patients, which is FLT3, ITD, and TKD, NPM1, and reflex to CBPA, you will have to almost rerun all your specimen for IDH2 testing to figure out who would be possibly eligible for this drug. So back to where the story started. In 2011, Dr. Andrew K. Godwin moved to University of Kansas Medical Center and started the clinical molecular oncology laboratory. And in 2012, our lab is CLIA certified. And in 2013, we get our first CAP on-site inspection and get accredited. And in 2015, we get our second inspection. And in 2016, we launched our first NGS testing in the lab. And here comes my job security. So we got, in our most recent inspection, we get 0 deficiency in molecular pathology. The inspector spent a whole day and flipped through my validation and our records. It's like this much just for validation data, summary, and protocol. And he finally gave up. He said, I couldn't find anything, so I'm just going to issue you a 0 deficiency, which we are very happy. To get there, what we need to do for a new test is we need to consider lots of things. First thing is why we are offering a test. Is there any clinical indication? Second, we have to determine the method. We need to search the market if there is any FDA-approved assay available. If not, is there any RUO research use only kits available? If yes, we find RUO kits, which manufacturer we are going to choose and why we choose that. And if not, that's the last choice. Can we develop a new assay in-house? For NGS I would say there is almost no way for a lab to develop the whole thing. The reagents, the bioinformatic parts is way more complex than a lab can handle. And for when we decided what kit we are going to do, then we need to do the assay validation. So we need to decide which instrument we are going to choose, how much calibration we need, and how much maintenance we need for the instrument, and the service contract, how much it's going to cost. Also, how accurate, precise, how reproduce, and what is the limit of detection, and also what is the reference range for the assay. These items all needs to be accurately validated. Also, we always have to pay attention to cost, like the cost, because we have to be able to move the lab forward. So unfortunately, we have to think about the cost. And after the assay is launched, we need to think about the QM, the quality management, QA, the quality assurance, and the QC, quality control procedures. These are really important, and these are things could get you in trouble and shut down the whole lab. And also, it is very important to have a very high quantity of these policies in-house so we can provide the patients the most accurate results. And last but not least, if you think you don't have any other exams after you get your PhD, I'll tell you that's totally wrong. And you got your continuing education, you have to get credits for your certificate, and also you have to participate in some kind of proficiency testing program. So our lab is participating with the CAP PT program. What does that mean is every 6 months we get a big exam, and if I fail, I probably will be kicked out. And so far, we participated twice with the NGS PT testing, and I'll talk about the results later. So the following assay validation we performed in-house is complied with the guidelines released in 2017. So you might have questions. I just said we launched our first NGS testing in 2016, and how can you comply with the guidelines released in 2017? So here, I have to say that you got to have some visions, right? When you are doing this, when there is no regulation, you have to guess what are those big guys over there will do later, so you don't have to redo your validation. Luckily, when we received or downloaded the guidelines, I was checking item by item, see if we are in compliance or not. So when I look at the requirement for analytical weight bench process, I said, okay, good, we've done that. And the analytical bioinformatic process has to be validated as well and check. So last but not least, the interpretation and reporting process has to comply with all the guidelines. And thanks for the QCI system we are using in-house, we are in compliance. So good for the lab, good for me, and good for the patients. So the next generation sequencing assay validation is a very complex and time-consuming process. Since I joined the lab in 2014, it took me about 2 years to get this validation done. This is simplified workflow. Briefly, we'll break the sections into 6 different categories, and I will use one of our NGS testing panel, hematologic neoplasm targeted gene panel, as an example. So per CAPS checklist for the NGS validation, we need to validate the DNA input, and we need to validate the library procedure. We need to do library QC, and we need to assess the NGS run on instrument. And after that, we need to validate the data analysis, and at the end, the outcome is a data interpretation and how we are going to produce a actionable, readable clinical reports. First, the DNA QC. I want to say, based on our experience, the DNA QC for blood and bone marrow extract DNA could be optional because we do run 80 samples, and above 98% of the DNA extracted from blood and bone marrow has very high quantity. However, this is not included in our heme panel validation, but based on our other experience for other panels for FFPE extracted DNA, we would highly recommend you have a DNA QC step, because based on our data, we have 86% high quantity QC samples, and 14% of the samples failed the QC. And during the validation, we proceed no matter what. We just want to see how accurate this prediction is. And it's 100% when you fail the DNA QC, you will not get a good library prep. And all the following QC will falling apart. And for the library prep, we're using the KISEC targeted DNA panel. I listed a couple of the reasons why we choose this panel. We also assist the other panels as well, but compared to other panels, this panel requires as low as 20 nanogram input, and it uses enzymatic fragmentation, so you don't have to buy the Coaris instrument. And this panel has a unique chemistry and method, which include a molecular barcode, and it will theoretically eliminate PCR duplication and amplification artifacts, so we can confidently detect low frequency variants. And this panel uses dual adapters. That means you can pull up to 96 samples in one MiSeq run. And the panel gives you a very universal application for PCR region, for DNA regions such as CEBPA. Those ones are hard to be amplified, but this was using this method we were able to. And I also want to mention this workflow is very fast compared to other methods. We were able to, our technicians can prepare the library within 1.5 days. And I am the champion in the lab. I do it within 1 day. So, but you can't ask your to do the same thing because I don't have other things to do. I can clear out all my tests, right, and just focus on NGS preparation. So my technicians, they have other duties to do. They have DNA extraction and other things, but they were able every time to finish the library prep within 1.5 days. And for the library QC, we use 2 methods per cap requirement. You have to do size check. We use a KiExcel, which is a fancy instrument that you don't have to pour gels. You can say bye-bye to agarose, and you do not have to fight who's going to do the dish, who's going to clean the dirty balance, and you don't have to calibrate your balance and everything else. But this assay is not enough to get a high, the accurate quantity. So you will have to use another PCR-based assay. We are using the KISEC library QC array kit, which has the standard preloaded. And you might think we are so lazy, we're trying to get everything automated and as much as reagents preloaded in the plate, so we don't have to do that. I would say deeply done, yes, a little bit, but more important is we want to improve the turnaround time. Each step can save us like a couple hours, which adds up to days. So that's the fundamental reason why we are choosing this automated instrument and these array kits. And here you can see that using the library QC, we get really nice even loading for our libraries, and our intensities are perfect. Our cluster density is around 1,000, which is optimal cluster density, and our QC scores are always above 90% using this panel. And the next 2 steps, I would say, is the limiting step for data for NGS validation. The first limiting step is bioinformatics part, how can you convert your FASTQ files to VCF files? How you call the variants is really difficult. And I was self-taught myself for a couple years, and I also talked to Kaigen's technical support almost every day. That's how they identified me as a speaker, because I bet everybody there, they knew me, because I just like, hey, it's me again, so question. However, after a couple years, I could see, actually, once you understand how to use the bioinformatics genomic workbench, it's a very powerful software. It's not very easy for users has no experience, but once you get to know how this works, it's really nice. I was able to configure our customized panel using their tools, and I also has all the output files for me for next analysis. Like I have VCF files I can use for QCII, and I do have other files I need for my QC documents. So last but not least, I want to talk about the QCII. Why we are using the QCII system? Because you will need a database to do this interpretation, and it's almost impossible for a small lab to do it by yourself. So the QCII system has all the support in the background, and the next speaker will talk more in details about this. I just want to quickly show you how it looks like in the system. You can see this is the surface. You will see all your variants are listed there, and computer already classified the variants for you. But you are going to go into the detail, click each of them, and review all the evidences and decide if you agree with this or not. And this system also gives you other information like drug treatment, clinical trials, and after you assist every variant, you just click there, generate the report, and you get your customized reports there, which is very nice looking, and our physicians are impressed by our effort and work. So in summary, we were able to implement NGS tests and achieve zero deficiency in molecular within 2 years. And this NGS library prep workflow is very comparable for different panels. We were able to pull samples together, which dramatically reduced the cost. And we generated a unique molecular database for patients and KUMC, which is much appreciated by our physicians. And also, in all, we improved the patient care we offer at KUMC. We have faster turnaround time. We have more possible treatment plan, clinic trial options. We provide precision medicine, offers higher quality of life, and the patients are satisfied. And some patients come to our center because we can offer NGS testings. So I want to thank you, my boss. He's supposed to be here. He probably missed my talk. So Dr. Andrew Gottman and Dr. Tilzer is our medical director, and all other people in the lab. Well, thank you. I'll take questions after all the talks. Thank you.
QIAGEN.
Sample to insight.